Hi, my name is Min, and I'll be talking about the effect of inhibitors on phosphatase enzyme and its optimum pH. The table of contents as follows: abstract, introduction, materials and methods, results, discussion, appendix, and reference. A letter of transmittal. All organisms have to break down into small to absorb what they need. This process is regulated by catalysts called enzymes. Enzymes accelerate the reaction without changing their characteristics. Most enzymes are specific that they only act on certain substrates. For this reason, substrates have to bind to special site on enzymes called the active site. Enzyme activity can be varied by the response to the environment, such as temperature and pH. Increasing temperature causes more collisions between reacting molecules, and therefore, the reaction increases. However, further increases may alter the conformation of the enzymes by disrupting the weak bond. In order for organisms to function properly, enzyme activity needs to be regulated. Inhibitors are the ones that slow down the reaction rate. Inhibitors are divided into two groups, irreversible and reversible. The enzyme used in this experiment is phosphatase. It is an enzyme that removes a phosphate from the substrate, which is the process called dephosphorylation. Phosphatase enzyme can be subdivided into acid or alkaline depending on the pH at which they exhibit optimum activity. The purpose of this experiment is to find out the optimum pH where the rate of phosphatase enzyme reaction is maximized. Also, there are 3 different inhibitors, and each is determined by the rate of reaction, whether it is competitive, non-competitive, or controlled. This report was created following the experiment conducted at Pennsylvania State University, Biology 230, Spring 2010. The experiment followed the instruction given by the course instructor and is based on the formal lab report created by the author. Abstract. Objective is to study optimum pH where the rate of phosphatase enzyme reaction is maximized in terms of 3 different inhibitors. Participants. This experiment was carried out by undergraduate students at Penn State University under supervision of many different TAs and Professor Ken Feldman. The evidence data collected from the experiment. The brief procedure is that each cuvette was set with 5 different concentrations of substrate. Students were divided into 3 groups. Each group did control, low inhibitor, and high inhibitor. Low inhibitor was 10 millimolar and high inhibitor was 20 millimolar. They were also observed for 2 to 3 times with 30 seconds intervals. The conclusion was that the data of pH and reaction rate showed that the maximum rate of phosphatase enzyme was when pH was 9, which is the optimum pH. From this result, it can be concluded that the enzymes used in this experiment is alkaline phosphatase since it is higher than the neutral pH, which is 7. Further changes in pH over the optimum show that the rate of reaction decreases, and it follows the concepts of enzyme activity mentioned in the introduction. Introduction. Enzyme activity can be varied by the response to the environment, such as temperature and pH. Increasing temperature causes more collisions between reacting molecules, and therefore the reaction increases. In order for organisms to function properly, enzyme activity needs to be regulated. Based on the primary mechanism by which they inhibit the enzyme, there are 2 mechanisms which are competitive and non-competitive inhibitors. Competitive inhibitors bind to the same active site as the substrate, so they can be overcome by increase in substrate concentration. It is reversible. Non-competitive inhibitors bind to another location outside of the active site and change the shape of the enzyme, so they are not affected by increase in substrate concentration. It is irreversible. Most organisms and tissue produce specific phosphatase, and their presence or absence is used for medical purposes. Besides, there are also practical uses such as cow's milk. Phosphatase levels indicate if milk is properly pasteurized or not. Low alkaline phosphatase level indicates it is properly pasteurized. High alkaline phosphatase level indicates insufficient pasteurization, and high acid and neutral phosphatase level indicates it is contaminated with microorganisms. The purpose of this experiment was to find out the optimum pH where the rate of phosphatase enzyme reaction is maximized. Also, there are 3 different inhibitors, and each is determined by the rate of reaction, which is competitive, non-competitive, or control. The hypothesis of the experiment is that if the enzyme is an acid phosphatase, its optimum pH is lower than 7, and if it is an alkaline phosphatase, then its optimum pH is higher than 7. The reason is that an acid phosphatase and alkaline phosphatase function optimally at acidic and basic pH, respectively. Materials and methods. A spectrophotometer was used to measure the amount of light observed, which indicates the concentration of product formed by the phosphatase enzyme. By the equation of Beer-Lambert law, observance equals ECL, where E is extinction coefficient, C is the concentration, and L is the light path length. The amount of color change over time can be measured, which is also the amount of phosphatase activity. By testing enzyme activity in cuvettes varying pH, we determined optimum pH. For each cuvette, each different pH was given from 3 to 10, and they were observed for 2 to 3 minutes with 30 seconds interval. The optimum pH was the pH where the rate of reaction was highest. For second activity, each cuvette was set with 5 different concentrations of substrate. Students were divided into 3 groups, each group to control, low inhibitor, and high inhibitor. What my group did was control, which does not contain inhibitors. This is the first result. Table 1 shows the pH and the rate of reaction. It shows the pH increases from 3 to 10, and the rate of reaction increases but decreases at the end. Figure 1 shows the reaction versus pH. It shows the rate of reaction with pH from 3 to 10 on the x-axis. pH 9 has the highest rate of reaction, and it decreased with further increase in pH. From data table 1 and figure 1, the rate of reaction starting from pH 3 to 0 increased continuously to pH 9, but suddenly decreased after that. Table 2, the rate of reaction with different substrate concentration. It shows that inhibitor makes the rate of reaction 0. However, as the substrate increases, the rate increases too. Figure 2, Michaelis-Menten plot of rate of reaction versus substrate concentration. It shows rate of reaction in 3 different conditions, which are control, low inhibitor, and high inhibitor. The control group had the highest rate of reaction, and high inhibitor had the lowest rate of reaction. Figure 3, Lineweaver-Burke plot of inverse rate of reaction versus inverse substrate concentration. It shows the inverse rate of reaction in 3 different conditions, which are control, low inhibitor, and high inhibitor. High inhibitor brings the inverse rate of reaction the highest, and the control group has the lowest inverse rate of reaction. Discussion. The results of graphs show that the inhibitors reduce the enzyme function and increase for the reverse reaction plot. The reason Lineweaver-Burke plot has only 2 points for low and high inhibitors is that some reaction rates are 0. Since the rate of reaction is 0, the rate of reverse reaction 1 over V cannot be come out. Overall, as the substrate concentration increases, the rate of reaction also increases. The data of pH and the reaction rate show that maximum rate of phosphatase enzyme was when pH was 9. Further changes in pH over the optimum show that the rate of reaction decreases, and it follows the concepts of enzyme activity mentioned in the introduction. Enzymes are essential in human body, but they do not always give positive effects. Their reaction rate should be regulated, and too high or too low reaction rate causes disease. For example, one enzyme called angiotensin converting enzyme works with kidney. If this enzyme is not regulated and highly active, then the renal disease comes in, eventually type 2 diabetes. On the other hand, for example, deficiency problem, neurogenetic enzyme can be an example. Loss of this enzyme activity causes mutations in the aspartoacylase gene, and finally the Canavan disease, which is associated with brain degeneration. Common symptoms known so far are mental retardation and seizures. Although there is no specific treatment that has been proved yet, current treatment is based on a combination of physiotherapy procedures. There are no specific errors in this experiment, but one thing might vary the optimum pH level. When the cuvettes were set into the spectroscopy, fingerprint might have been remained and observance of light could not be perfect. Since they were just checked by eyes rather than running some tester, it might cause a slight difference. This is the appendix: Beer-Lambert law, observance ECL, E extinction coefficient, C concentration, L the light path length, and this is the references.